So then I'll get started. All right. Well, thank you very much for coming. It is my great pleasure to talk to you today about tomatoes. And it's going to be slightly information dense, so I'll just jump right in. And I guess I'll fast forward right away to what's normally the closing slide, because what I am talking to you about today is, of course, not at all my own work. I mean, I'm gluing things together here and interpreting and doing some of the technical stuff. But in a study like this, you know, it takes a village to raise a child, and in this case, you know, the results I'm showing you today would not be possible without the contributions of all of the people shown here, who are affiliated to the various institutions shown in the top right. And as the talk progresses, I'll kind of point out also the various contributions. So this is just to acknowledge that this is obviously teamwork. So what are we talking about? Well, what can a 500-year-old tomato actually tell us about domestication and what I'm going to call the Colombian exchange? And this particular 500-year-old tomato might be known to many of us, and also this particular photo, because this is, of course, one of our most cherished specimens. And this photo, which Ronald will see soon as he can go back to the institution, is also used as in the wall art in some of the offices. So we all kind of know what this is about. Now, there's also wild tomatoes, and so what I'm showing you here is the larger clade of tomato-like things, and there's no prizes for guessing which one is the domesticated one. Now, but in the top row, you can see a couple of other ones that are closely related, and they are, that group is termed very easy to remember because they're known as the yellow or red fruited tomatoes, which is a little monophyletic group within this larger clade. And among those, the Solanum pimpinellifolium is actually the wild ancestor of domesticated tomatoes. Now, the story of the domestication is a little bit contentious for a couple of different reasons. One of them is that the center of diversity for the wild ancestor is in the Andes, and kind of going by the traditional logic of Vavilov and others, that would place the center of domestication also in South America, in the Andean region. But there's also other evidence. For example, there's actually in Mexico a really rather old weedy feral tomato, so something that may have been kind of like had a longer running relationship with humans, let's say, and was around there, and seems to have been the source population from whence tomatoes were domesticated, and by recent reconstructions, actually a rather long time ago. 7,000 years old is what's being mooted in a recent study, and that's pretty old for any domesticated species. Now, another thing that's a little bit contentious is the relationship between three different things, here shown at the bottom. There's the Solanum pimpinellifolium, the wild ancestor. Then at the top is Ceraciforme, so cherry-shaped, or cherry tomatoes, and Lycopersicum, the big tomato. And kind of a naive model for the domestication of tomatoes holds that the cherry tomato is like the intermediate. Well, the evidence that we're going to look at doesn't show a particularly clear picture about that. Some cherry tomatoes are intermediate, others are just like small tomatoes, because, well, fruit size can be arrived at in multiple ways, right? So a little bit complicated, but anyhow, tomatoes were domesticated in the New World. The name tomato comes from tomato, which is a word from the Aztec, the language of the Aztecs, who of course had this big pre-Columbian empire in central Mexico. But then in 1492, Columbus sailed the ocean blue, and it started off one of the biggest events in our history as a species, which I'm going to call here the Colombian 1492, and was an event where people and ideas and goods, oftentimes not voluntarily, were exchanged between the old world, Afro-Eurasia, and the new world. As we all know, especially regulars of the colloquium, this is the impacts socially and botanically of this whole event are one of the topics of study of our colleague Tinde, who recently gave a very interesting talk about the introduction, going the other way, of West African rice into Suriname. Anyway, during the Colombian exchange, all sorts of interesting neotropical crops found their way to Europe, and there they became curiosities for Renaissance Europeans, who started making herbariums, among which the En Tibi perpetuus ridentem floribus hortum, which is an ongoing topic of research by both Tinde and Anastasia. And they have, you know, as the insights developed, the date that this was produced is being pinpointed at probably 1558. So this is what it looks like on the outside, and the title, well, I've seen several translations from the Latin. I guess maybe it's something like, "For you, an eternally smiling flower garden." And so probably made in 1558 in Italy. It contains many different plants, but also tomatoes. And next to it, there's an annotation that says, well, this is a love apple, but I'm not quite sure if that might be a scribal error, but okay, Tinde knows much more about this, and Anastasia too. Anyway, so this book has then a very rich and complicated provenance all throughout Europe to finally end up in Leiden, and it's now one of our most cherished objects. Now, a couple of years ago, our colleague Barbara started advancing this concept of museomics, which is basically the idea that we might be able to unlock more of our collection materials using ancient DNA techniques and high throughput sequencing. And this idea got some traction, so much so that the En Tibi tomato specimen has also been sampled destructively, as you can see here. So a tiny little bit of leaf material was taken from it, and to extract DNA out of it and sequence that. And so that happened. Of course, this was all 500-year-old DNA, so be a little bit careful with that. And then subsequently, it was sequenced in Wageningen by our colleague Elio Scheile, or just using what's one of the kind of standard platforms for high throughput sequencing, the Illumina HiSeq 2500, which has read lengths of 125 base pairs in an ideal world. But then, well, let's try to assemble it. And so here's how that goes. Among tomatologists, the standard tomato is the ketchup tomato Heinz 1706, which is an inbred cultivar that was sequenced to build the reference genome of tomatoes and annotate that to find all the genes. And so this is basically the North Star. If you do any tomato research, this is what you orient yourself on. And here too, we mapped our reads against that reference genome. Now, unfortunately, that then looked like the figure that we see here in the top left. So this is the first roughly 1,000 bases of the much larger genome. And you can see that the coverage is very low and very uneven. So there's whole stretches with nothing or only a couple of reads. The other thing that we can see is that the DNA is fragmented to such an extent that the bits that remain to sequence are even shorter than the short read lengths of this platform, which is 125 bases, right? Because what you would normally expect to see is an assembly that is, let's call it staggered, so it kind of neatly lines up. But in this case, it is more jagged, so it doesn't neatly line up. And okay, that's kind of a bummer in itself. But it also means that after this, or subsequent to this whole fragmentation, there's a lot of exposed tail ends that have been, facing the elements for the last 500 years. And one of the things that then starts happening is a type of biochemical decay known as deamination, where a cytosine becomes uracil, we might know from RNA, right? And then the sequencing device interprets that as thymine, the letter T. So if this process is happening, then what you would expect to see, unfortunately, is that the tail ends of each of your reads is going to have an excess of thymines being detected. And the plot on the right shows that this is also the case for the En Tibi specimen. So what we're looking at there is basically analysis that integrates across all of the millions of different reads, and it looks at what is right at the cusp of, the threshold of where that read begins or ends, and that's shown as the relative position. So as the numbers on the x-axis become positive, we start reading into the actual fragment, and you can see that right at the tail end, there's this peak in the red curve, which shows indeed there is an excess of thymine. So there's fragmentation, there's deamination, and there's very low coverage. So that was all a little bit disheartening, because the promise of actually being able to recover full gene sequences and do functional genomics on plant traits, well, that unfortunately went out of the window. But nevertheless, science progresses, and in the subsequent years, at least 2 papers were published, which were inspiring and useful in this project. First one here shown on the left is actually a preprint of our colleagues at the Museum of Natural History in Gainesville, Florida. And there they had a problem because they had some butterfly specimens in their collection which were not geo-referenced. So they couldn't know where exactly they'd been collected. But they also had a whole bunch of other conspecifics of that same butterfly that were geo-referenced. So they came up with a kind of clever approach to triangulate basically where the unreferenced specimens must have been from, just depending on just based on sort of their affinities with the other georeferenced specimens. And obviously they could only do that because they had kind of a good panel to compare against of other specimens that are georeferenced. Now, the next nice piece of the puzzle that then fell into place was this study shown on the right, where a whole bunch of tomato genomes have been sequenced, 360 of them, including many that are land races and local cultivars from the New World, and which are georeferenced. So in principle, it should be possible to use a similar technique as in paper 1 to kind of recover where genetically the En Tibi tomato is from. Now, that required a little bit of complicated bioinformatics to reconcile this fragmented genome there, take out just the bits that have sufficient coverage. Here, okay, sufficient is 10 times coverage. Take those coordinates, then go into the SNP data sets of these 360 tomatoes, look in those same coordinates, and then bring it all together in a relational database, yada, yada, yada. Complicated, but then there was data to compare. And so let's look at some of these results. So the first thing that we did was do a bit of a dimensionality reduction. Now, I previously said there was very little data, or low coverage, fragmented, and that's the case. Now, the SNPs that could be recovered with sufficient coverage, that's still over 8,000 in the En Tibi, and the stretches of the genome that had sufficient coverage, if we then look in all these other tomatoes, we still get over 250,000 different variant sites. So think of this as like a character state matrix that has over 250,000 columns, each of which are variable. Now, that is, of course, a highly dimensional data set. So the first step was to reduce that dimensionality. And here I used a data science technique that I'm told is pronounced as TSNE. which it was invented by a Dutch guy a couple of years ago, who is now absolutely swimming in the big bucks, working for Facebook, I think. But the algorithm is open source, and I ran it on our data, and you kind of need to think of it as a bit like a very hip type of principal component analysis. And what it shows is, well, indicated with the red arrow, the En Tibi specimen, which falls within a sort of a cloud of both big tomatoes in blue and cherry tomatoes in the open shapes. And then to the top and kind of left is most of the Pimpinellifolium accessions. And then there's two more species that neatly cluster with themselves, shown in the bottom and more to the left in the bottom. So, okay, first piece of information, the En Tibi tomato is a tomato and not maybe something wild or something else. It is basically a normal tomato. Now, when we look at the shape of these different markers, the diamond shape is Mesoamerica, so Central America, and then the squares are South America. Unfortunately, there's not a clear geographic signal here. Okay, zooming in a little bit, we then did a network analysis. So here we're just looking at the complex of the wild ancestor and cherry tomatoes and big tomatoes. And we can tell these apart by the different circles here. So we're looking at a network and not a tree, because these are all things that readily hybridize. In fact, the larger clades, especially the orange-red fruited tomatoes, they all just readily hybridize. So there's gene flow all between them, and we're here looking at just nuclear DNA. So there's also this recombination. And the way then to display the diversity is obviously through a network and not through an acyclic kind of graph. Each of the circles is specimen, and the IDs that we're looking at, the first letter shows what's what. So P is Pimpinellifolium, and you can see that they are mostly concentrated on the left-hand side of the plot. Another thing that you will notice is that the branches leading to each of these are relatively long. So you can interpret this as showing that the genetic diversity within natural populations is generally larger than in domesticated crops, which are more inbred, as shown here. Then moving to the right, we see more things starting with C for cherry, and there's some clusters there that show it to be kind of intermediate. And then on the right, there is kind of a mess, which is the big tomatoes and the cherry tomatoes. And they are also color-coded by geographic origin, as you can see. And most of the Mesoamerican accessions are, again, in that little hot mess on the right-hand side. And we'll zoom in on that. Last thing here to notice is these little pie charts on each of the nodes. I'll explain what those are in a minute, but it's kind of worth noticing that there's three different shades, black, gray, and white. And most of the pies on the left-hand side are black, and then moving to the right, there's gray and white popping up. Right, let's zoom in a little bit. So now we are looking at that little cluster that was on the right-hand side of the network on the previous slide. And here we can then see in black the En Tibi tomato, and we see its nearest neighbors, 249, 153, and 233. And their origins are here shown on the map to the right. So it is quite appealing to interpret this as giving some indication of where the genetic origins of the En Tibi tomato lie, namely in southeast Mexico. Kind of looks like that. Another thing that you will notice here is that in this case, all of the little pies are solid gray. Okay, so what's that mean? This is another way of looking at the results of a structure analysis. So this analysis was developed by my student Ewout, who, well, what does it do? So each of the slides in the plot here is an accession. And then what the algorithm is trying to do is it tries to assign all of the SNPs in that specimen, each of them to an ancestral population. And then the algorithm basically does a parameter sweep where it first looks at, well, can everything be assigned to one ancestral population? Or how about two? How about three, four, etc., etc.? And then it obtains the maximum likelihood estimate, which in this case is 3 ancestral populations, red, green, and blue. And so here on the right-hand side of the plot, we can see the En Tibi tomato, indicated with red arrow, coming from the blue ancestral population. So this corresponds with the gray solid pies in the zoomed-in plot that we were looking at on the previous slide. So that's kind of neat that that group is also reconstructed as all being derived from the same ancestral population. I see that as kind of confirmatory evidence side by side with the network plot. Okay, so origin, population structure. One thing that this also shows is that there's clearly no neat sorting of the different things. Cherries, big tomatoes, Pimpinellifolium in distinct ancestral populations. Right? There's 3 ancestral populations. They don't map onto these 3 things. So anyway, so that kind of shows that a very simple model where the cherry tomato is an intermediate. That may be true for some of them in the network, but others they're just small tomatoes because, well, you can breed big tomatoes, you can breed small tomatoes. Right. Anyway, what else can we learn about this whole process of domestication? Another analysis that Ewout did was to look at the heterozygosity. So this is where you look at each of the individual variants across the genome, and you look at whether those are homozygous. So are both copies different from the reference genome, or are they heterozygous? And then that could be heterozygous in a couple of different ways. And here the En Tibi tomato is noticeably more heterozygous than the cultivars. And in fact, it falls a bit more within the range of wild tomatoes shown here on the left. And here on the right, you can see also that the specimen compared to the other Mesoamerican tomatoes is very much more heterozygous, or in other words, much less inbred than the present-day cultivars. So what can we conclude? Well, being a little bit conservative here, I would say that the En Tibi at least shows genetic affinities with tomatoes currently found in southeast Mexico. But, caveat, humans and their crops move around in ways that are totally different from sort of the natural diffusion where genetic distance and geographic distance normally kind of correlates. You know, humans develop trade lines, conquer new territory, etc., etc. And so the geographic patterns are obviously different. So it's kind of hard to triangulate, but okay. In any case, the evidence that we have so far suggests Southeast Mexico. What's interesting, second point, is that the specimen is much less inbred than present-day tomatoes. So it does show that there is genetic diversity that we no longer see. And so that's kind of tantalizing in that there could be lost traits in this tomato, but due to the fragmentation, with the current stage of the technology, we can't do a lot of functional genomics to actually interpret what those differences are and try to recover full gene sequences. But there's at least evidence that a great deal of diversity has been lost. And then the final point kind of ties back to that thing about the cherry tomatoes. Then, yeah, some of them might be an intermediate between Pimpinellifolium and the big ones. Others are not, but we kind of knew that already. So, okay, the nuclear DNA that we so far have been able to look at, because that's what been produced and published by this 360 tomato consortium, is very fragmented. But there's also chloroplasts, and in every plant cell there's way more chloroplasts than nuclei. And so, as a very recent development, I did a quick assembly to map the En Tibi data against a tomato chloroplast ref-seq. And shown here on the left, you can see that the coverage is rather good. And so we just see a little part of the chloroplast, but as you scroll through it, it basically looks like this all throughout, which is very nice. So what's also nice, it's non-recombining. So we could maybe zoom in a little bit more. Downside is that there's not really a good panel to compare against because, well, the 360 tomato project didn't publish any chloroplast data. However, the raw data is out there, so in principle, you could just remap all of those against a ref-seq and then recover chloroplast markers in that way, which is precisely the idea. So this is the project that Vasia here shown in the picture is starting out on for her master's. So, okay, there's future work here to do. And then I guess to close off, there's also a little bit of past work as a displacement activity during last year's lockdown. I ordered some seeds from the seed bank at UC Davis of the nearest tomato that is georeferenced with lat-long coordinates nearest to the En Tibi, which is a little bit further away though than these Mexican ones. And I figured, well, let's give it a shot. And it turns out that tomatoes can get quite big. So you can see here how my living room was gradually being taken over. But finally, they bore fruit. And as a true scientist, I, of course, then took measurements in the size, the weight, also the color. And you can see the tint resembles my kitchen cabinets quite closely. I did a taste test, and it's although it really looks like Albert Heijn tomato, it actually has fewer of that like sort of soggy, slimy stuff in the center. It's quite meaty and slightly more umami, so quite tasty. Heartily recommend it. So I guess this brings me to the end of my presentation. So. Thank you very much for your attention, and I'll be happy to take your questions.